a big aim of our research is to try to understand how plants fabricate petal patterns. If you've been in a garden center or in a field, you will have noticed that there is often colorful mark on the surface of petal. And this is what makes flowers particularly appealing to us. We like their scent, but we like how they look. They look pretty, but they don't look pretty for us. We know they do many things, and one thing they do is to help plants talk to the animal that pollinate them. So this is very, very important, because without this pattern, probably pollination wouldn't be as efficient, and we would struggle to produce seed and maintain the next generation of plants. And we think actually this may have play a big part in why flowering plants have been so successful. But we still don't know how plants fabricate this pattern, and we also do not know how evolution can play with the different processes to create the diversity of floral form we enjoy today. This simple pattern is what we call a bullseye. It looks like a target. Basically all you need is for the petal to have two regions that contrast with each other. So here we have the base of the petal is purple, so I make the center of the flower dark. And a periphery of the petal, what we call the top part, is white. And this creates a contrast. To understand how these patterns are made, we need to understand how does the flower know to divide the petal into two regions. And for this, really, we had to combine tools from developmental biology, genetics, evolution, and computational modeling. So SLCU is perfect for this kind of work because we really needed to combine both experimental biologists and computational people. And what we try to understand is, how do the cells know where they are? When does the petal become divided? Lucy went and dissected very, very tiny buds. And when we dissect them, the petals are very small, less than a millimeter tall, and they're completely green, they have no pattern. So to us, they look like unpatterned petals. But through the pipeline that Lucy developed, we realized that the cells already know where they are. So there is already a division between the patterns. So if you want to understand how flowers make pattern, we need to look much earlier, because basically flowers plan early. And they create a template for this pattern a long time before the pattern becomes visible to us. What we knew is that this species of hibiscus belong to a group that's widespread in Australia and New Zealand, and the plants vary a lot in the term of the size or the color of their bullseye pattern. So for us, it's interesting to understand how evolution creates this diversity, because potentially, when you change the pattern, you can attract different pollinators, meaning that the plant can become isolated and you can create new species. So it's really a way of understanding where biodiversity comes from to begin with. And what we realized is flowers may look the same, but it doesn't mean that the patterns are made in the same way. We found at least two ways of creating different sizes of bullseyes. The first way is simply drawing the line, what we call the bullseye boundary, so the separation between the two domains, to put it higher or lower in the petal early in development. That would be a change in the patterning process. But we found out that it was also possible for the plant not to do that, not to change anything in the patterning, and instead change how much the petal is growing below or above this boundary later on. So you can end up with proportion of the bullseye that are the same, but they have been produced completely differently. And for us, it's important because it means that evolution had many tools in the toolbox to create diversity. If there was only one way of doing things, first it would be boring, and also it would probably make it very complicated. But because there is many, many ways that evolution can use to change the proportion of the pattern, it probably explains why it has been so easy for evolution to generate diversity.
So we have demonstrated how you establish an early boundary, and what are some of the developmental mechanisms allowing to vary the bullseye proportion. But does bullseye size actually matter for pollinator attraction? So first, we tested the behavior of bumblebee in our bumblebee lab and tested if they had an ability to distinguish between flowers with different bullseye size. To do this, we designed an artificial disc with different size of purple center that mimics the size of our real flower. And what we found is that bumblebees were able to see a difference between those two bullseye size, but they were also having a preference for a medium-sized bullseye when they were compared to a smaller bullseye. We also compared the medium-sized bullseye to a larger bullseye, and what we found is that bumblebee didn't have any specific preference. They were 50-50% attracted by those two bullseye size. So we then tested if bullseye proportion had an impact on flower detectability. And to do this, we calculated the bee flying speed between three discs of the same type, placed at equal distance from each other. So we recorded their flying speed and measured the time they spent flying between them at the computer. And what we found is that they were actually flying 25% faster between artificial discs with a medium-sized centers compared to the smallest one. So this suggests that actually a larger bullseye makes the flower quicker to detect, And it may not sound a lot, but actually, in the same amount of time, bee can visit 4 flowers instead of 3. So this can make a difference when we know that actually flying costs energy for the bees.
So there are many things we can do from this research. First, technically, this had allowed us to develop pipeline and tools that are now going to be used widely for us to characterize genetically modified line, but also natural variants. So this is really useful for us. There are 2 main questions we want to answer. One is a very fundamental developmental question. So what the data of Lucy show is that there is very likely a signal, a mobile signal, a small molecule, we don't know what it is, but that come from outside of the flower. It could be the center of the flower, it can be a different part of the plant, that's going to circulate in the petal and position this boundary. And for us, we really want to understand what is this signal, what is the process here, especially because from an evolutionary viewpoint, we think that petals are modified leaves, and that there is a very good chance that this patterning mechanism already exists a long time ago, before flowers were invented by evolution, to pattern leaves. It's just that leaves are far less showy. They are the quiet lateral organ, and it's much harder to pick up on potential defect and phenotype in leaves. So we think that with petal, we have actually a very good system to pick up, to identify genes and processes that are important to pattern all lateral organ of plants. So that's the first thing that our lab is going to do. The second thing is, of course, we are only just starting to pick apart how pollinators perceive this pattern. So a big question we have is how different 2 bullseye need to be in terms of size per bee to pick up the difference. Is 5% size different enough? So that's what we are testing. The second aspect is the importance of the contrast. So what we know is a bigger bullseye is better than a small one. But to get a bigger and bigger bullseye, the plant needs to produce more pigment, which is energy consuming. And what we realize is if the bullseye keep growing, we will start losing the contrast. So maybe there is a kind of golden ratio, an optimum proportion that both minimize the amount of pigment the plant needs to produce, but at the same time provide enough contrast. So we want to test if that's the case. We want to see is there some kind of preferred proportion that maximize contrast. And also what happens if instead of having white and purple, we start having pink instead of white. So we diminish the contrast as well. So these are all things we are going to investigate in the future.